Hello friends, this is Dr. Sachidanand Singh. I am Professor of Biotechnology and I am happy to be part of this lecture series of protein prediction and analysis. So, in our previous two lectures, we have understood about protein structure prediction and second was protein structure prediction using softwares. So, in that software, we studied about modeler and then some online tools and databases. Now in this lecture we will understand the protein which we have modeled, how to validate that protein. So, in our previous lecture also n number of times a revision is done that for validation we have to use different methodologies using Ramachandran plot. So, now in a very clear identification and understanding we will see which software tools or databases can be used when we have a protein structure as a 3D structure, how it can be validated. So, let us start the lecture. So, just give you a brief overview what we have gone through the two lectures, that we saw that the single letter amino acids can be converted into secondary structure. That secondary structure helps us to wind up and put it into tertiary structure, and finally, they again get coiled and supercoiled to give a quaternary structure, so that they get compacted and perform their activity. A very short overview and brief that we have understood in our previous lecture, that how computational method helps in protein structure prediction. So, as you all, I am aware that you will understand that protein structure prediction is done by three methods, that is, and that also computationally is through homology modeling, ab initio and threading. And for that, in the previous lecture we saw that how homology modeling, the best possible method structure prediction can be done using our unknown sequence to target selection, template selection, then backbone generation, loop modeling, side chain modeling, model optimization, and finally model validation. So, today in this lecture we will see only model validation. So, now we have understood that in the previous lecture we have a PDB structure as a model optimized structure, either by completely done after MD or MC as well. So, this model has to be now evaluated or validated. So, for this model validation, the well known server is known as SAVS. So, let us type in Google SAVS, SAVES, and you can see under UCLA you will see SAVS as our one of the link. So, when you click that link, you can see it will give you a open user interface as presented in this slide. So, it is UCLA DOE lab and the present version is SAVS version 6.0. This SAVS version is taken care by University of California, Los Angeles, and it is run under their server platform. The most important version in this SAVS 6.0 which predicts the secondary structure are ERAT, Procheck and Verify 3D, and their references are been reported down. So, when you upload your optimized protein model which you have developed through different softwares or online databases, when you upload that in the previous slide as we saw the run program, when you select in the run program your protein, you can see the protein which we have taken, it has been submitted and the job ID is represented as #142 with .pdb extension. So, now phase by phase we can analyze each data, each online data category, and we can understand the result. Understanding the result gives us a clear picture of model validation, which is very good in representing into your papers for significant publication. The very first one is ERAT, second we can see Verify 3D. Probe, it has shown the server is not working that time when we started working for this protein. The fourth one is Procheck, which we will cover in another lecture also, and the final and the important one is Procheck, which obeys the Ramachandran plot. So, now let us see how this SAVs work. So, the main aim of SAV is these operating instructions describe how to run Procheck suite of programs for assessing the stereochemical quality of a given protein structure. So, this protein structure is under SAV's Procheck category. The main aim of Procheck is to assess how normal or conversely how unusual the geometry of the residue in a given protein structure is as compared with the stereochemical parameters derived from well refined high resolution structures. Unusual regions highlighted by Procheck are not necessary errors as such, but may be unusual features for which there is a reasonable explanation. Example, distortions due to ligand binding in the protein active site, and nevertheless they are the regions that should be checked very carefully in the another strategies. The input to Procheck is a single file, that is a PDB extension containing the coordinates of our protein structure which we have developed through the previous lectures which we have understood. This must be in a Brookhaven file format. One of the byproduct of running Procheck is that our coordinates file will be cleaned up by the first of the program structure. The cleaning up process corrects any mislabeled atom and creates a new coordinate file which has a file extension of .new extension. The .new file will have the atoms labeled in accordance with the IUPAC naming conventions. We all understand IUPAC. I have commissioned biochemical nomenclature which was given in 1970, which we have studied in our class 11th and 12th in chemistry. At present, hydrogen atoms are ignored, atoms with zero occupancy are omitted from the analysis, and only the highest occupancy conformations are retained whenever they are alternate conformations are identified. So, this is the input file for the Procheck, or we can say in the SAVs the input file has been given. So, you can see that Hoven format of the input of the Procheck, we give a PDB file into the SAVs, it itself convert into different format for the different upload. So, for Procheck it makes a format like this to give us the output. So, now let us see the output of SAVs or Procheck. The output comprises a number of plots, and these beautiful plots can be taken into presentations or report writing or project writing, and they explain with a detailed residue by residue listing. The plots are output as an PostScript format which can be opened into Adobe system, and so can be printed off a PostScript laser printer or displayed on a graphical screen using suitable softwares like example GhostScript or GhostView or Sun or a Silicon Graphic workstations, or PSView or Silicon Graphic Iris 4D system can also be used to see this graphical representation pictures. The plots can be in color or black and white and are described in sample plots, which we can see very clearly as different color backgrounds. So, let us now assume and understand the output of a Procheck. In the Procheck log file, you can see for that specific protein the chi square values, and for each amino acid the results are been given and reported. If we come down to the log file, we will be able to understand the percentage of all the amino acids are specified into which category. So, now in the log file you can see at the down for the one abc.pdb file which we have given as an input, the Ramachandran plot represents 89.9% of selected or allowed regions or core regions has been identified. So, this is an acceptable protein which has been reported, in which 9% around are allowed regions and generously allowed region, and very few are in disallowed region. As per like this, different factor analysis and bond length, bond angle, torsional angles understanding is also reported in the log file. So, as we go down in the log file, a very complete understanding can be represented and can be technically written into report or scientific paper. So, to explain this all with scientific component, understanding of Ramachandran plot is important. So, what is Ramachandran plot? In a polypeptide, the main chain NC alpha and C alpha C bond relatively are free to rotate. Hence, these rotations are represented by the torsional angles which are known as phi and psi respectively. G and Ramachandran used computer models of small peptide to systematically vary phi and psi with the objectives of finding stable conformation, and he reported under the 4 quadrant of phi and psi to fix these amino acid and report them as allowed or disallowed amino acids. G. N. Ramachandran also reported in his plot the phi value and on the x axis and psi value on the y axis. So, x axis is with the phi value and y axis is with the psi value. Plotting the torsional angles in this way graphically shows with the combination of angles are possible or not. In the pictorial representation, very clearly we can see that 4 quadrant place, and in which it represents the alpha sheet, left handed alpha helix and right handed alpha helix. So, plotting the torsional angle in this way will graphically represent the torsional angles of each residue in a peptide defines the geometry of its attachment to its 2 adjacent residues by positioning its planar peptide bond relative to the 2 adjacent planar peptide bonds. Thereby, the torsional angles determine the conformation of the residues and the peptide. Many of the angle combinations and therefore the conformations of the residue are not possible because of the steering hindrance, and we can see this steering hindrance happens in this phi and psi. The pictorial representation very clearly depicts the C alpha N bond where the torsional angle are developed at phi and psi, and the R group is represented at the adjacent. So, this is the Ramachandran plot of 4 quadrant where, as we saw in the previous slide, phi angle is the x axis and psi angle is the y axis. In the first quadrant, the yellow region is the allowed region and the red region is the acceptable core regions, which is the beta sheet, wherein the next quadrant is the left handed alpha helix and the third quadrant is right handed alpha helix, wherein the fourth quadrant nothing fits into it. The white portion is the disallowed region amino acid. So, any amino acid which comes into white region is not acceptable and the structure will not be validated or is not stable. So, in the above diagram, the white area as depicted corresponds to conformation where atoms in the polypeptide come closer than the sum of their van der Waals radii. So, now seeing the Ramachandran plot, we can see the Ramachandran plot shows the phi and psi torsional angles for all the residues in the structure except those at the chain termini. So, in that way glycine residues are the separately identified by triangles, as these are not restricted to the region of the plot appropriate to other side chain types. The coloring shading on the plot represents the different regions described in Morris et al. Paper as well. The darkest area as shown red corresponds to the core regions, as we already saw, representing the most favorable combination of phi and psi values. Ideally, one would hope to have over 90% of the residues in these core regions. The percentage of residue in the core region is one of the better guide to stereochemically quality approved. So, all the amino acids which are in the core regions justifies the best possible amino acid structured protein as the validated protein. So, the plot represented at the right hand side shows the Ramachandran plot of each 20 different amino acid types. The darker the shaded area in the each plot, the more favorable is the region. The data on which the shading is based on has come from a data set of 163 non-homologous high resolution protein chains chosen from structures solved by X-ray crystallography to a resolution of 2 angstrom or better and R factor no greater than 20%. So, the pictorial represent clearly depicts about it. The number in brackets following each residue name shows the total number of data points on that graph. The red number above the data points are the residue number of the residues in the question, that is showing the residues lying in the unfavorable region of the plot. The triangular points are the glycine which does not obeys the Ramachandran plot. Now let's see the another one, that is Chai plot. The Chai plot shows the Chai side chain torsional angle combination for all the residue types, and those side chains are long enough to have both these angles. The shading on each plot indicates how favorable each region on the plot is. The darker the shade, the more favorable is the region, which we can see. The data on which the shading region is based has come from a data set of 163 non-homologous same high resolution protein chains chosen from structure solved by X-ray crystallography. So, this data has been taken from a X-ray crystallography data to a resolution of 2 angstrom, and better R factor is 20%. And it is the same thing, the number in the brackets following each residue name shows the total number of data points on that graph, and the red number above the data points are the residue numbers of the residue in question, and they represent or depict the unfavorable region of the plot. So, now let us see the 6 graph representation. At the right hand side you can see the 6 graphs on the main chain parameter plot shows how the structure represented by the solid square compares with the well refined structure of a similar resolution. The dark band in each graph represents the result from the well refined structures. The central line is a least square fit to the mean trend as a function of resolution, while the width of the band on either side of it corresponds to variation of 1 standard deviation about the mean. So, you can see the dark band and the dashed line reported. In some cases the trend is dependent on the resolution, and in other cases it is not. This plot is intended as a rough guide only, and too much reliance should not be placed on getting results that are better than structures of the same resolution parameters. Yeah, so this one is a very important graph represented, and this graph we can see at the right handed side is a histogram graph. This histogram graph on this plot shows the distribution of each of the different main chain bond angles in the structure. The solid line in the center of each plot, you can see in the graph, we can see in the graph the solid line in the each structure of the center of each plot corresponds to the small molecule mean value of all the amino acid conformation structure, while the dashed lines either side shows the small molecule standard deviation. So, both the statistical calculation of mean to standard deviation is done. So, higher the standard deviation, the more is the distortion. So, it has to be concise together as mean and standard deviation. Highlighted bars correspond to values more than 2.0 standard deviation from the mean, though the value of 2.0 can be changed by editing the Procheck.prm file. So, it is user friendly that what level of standard deviation has to be kept. Default standard deviation kept is 2.0, but we can see or we can do a stringency by changing the standard deviation or increasing the standard deviation. So, if any of the histogram bar lie off the graph to the left or to the right, a large arrow indicates the number of these outliers, as on we can see CACO and CBCAC plots above, or which can be seen in the right handed side graph representation. The histogram on this plot also shows the distribution of each of the different main chain bond lengths in the structure. Bond length plays a very major role in the secondary structure prediction and validation. The solid line in the center of each plot corresponds to the small molecule mean value, while the dashed line either side shows the small molecule standard deviation. In the highlighted bars correspond to the value more than 2.0 standard deviation from the mean, though the value 2.0 above change the editing of Procheck PRN file. It is the same as we report in the previous histogram file, but if any of the histogram file lie off the graph to the left or right side, a large arrow indicates the number of these outliers. As in previous one we saw a CACO graph, but here we can see a CO plot above. So, these all previously reported graphs also gives us an understanding of few statistical parameters like root mean square deviation and RMSF. So, these histograms shows the root mean square deviation RMS distance from planarity for the different planars group in the structure, in the sense from the base structure to the developed structure how much deviation has been done. We can see with our own palm, if the RMSD is 0, the palm to palm matching is same, but if little deviation happens, the RMSD increases. So, that is the whole concept of root mean square deviation. The dashed line indicates different ideal values of aromatic ring. So, wherever aromatic rings are there, there are bit of deviation can happen because of the functional group. For example, amino acids like phenylalanine, tyrosine, trypsin and histidine, and for planar end groups like arginine, aspartame, aspartic acid, glutamine and GLU. The default values are very specific angstrom, that is 0.03 angstrom and 0.02 angstrom, which is very minute because the best possible conformation has to be reported. But again the same, the values can be altered by editing the what we saw in the previous one was Procheck.prm file. Histogram bars beyond the dashed line are also shown as highlighted because they become as a outlier. So, the options for this is, the main option for the plot are RMS distance from planarity for highlighting outliers for ring groups. Default RMSD is 0.03 angstrom. For RMS distance from planarity for highlighting outliers for other group, default is 0.02 angstrom. So, on the basis of this as a default, the plot can be seen either in color or in black white, which is been reported in the right hand side graph as well. So, now let us see these plots shows all the distorted main chain bond length, main chain bond angles and planar group. The torsional angles, or to understand the structure, bond length, bond angle, torsional angles are been taken into very nice consideration. So, the parameters defining how distorted these properties need to be before being plotted here are given in the Procheck.prm parameter files. For each main chain bond length and angle plotted, the plot shows the ideal value and the actual value and the difference between the two. So, three things are been reported: ideal value, actual value and difference between the two. For each distorted planar groups, three orthogonal projections are plotted and the value shown is the root mean square distance of the atom from the best fit plane. So, the options and the main option for the plot are: first, cutoff deviation from ideal bond length, cutoff deviation from ideal bond angle, bond length is angstrom, cutoff deviation from ideal bond angle is degree, cutoff for RMS distances and planarity, and final one is these plots can also be seen in color or black and white. So, till now what we saw was using UCLA SAVS analysis and seeing the different categories of what if, Procheck and Verify 3D, in which in this lecture we particularly specified Procheck all the graphical representation and the plots which can be analyzed. In this lecture, we have also seen that how Ramachandran plot basic concepts is utilized for Procheck analysis. So, during this whole lecture series of the present content, we have understood about Ramachandran plot secondary structure prediction and then using UCLA SAVS method specific Procheck analysis for torsional angle identification. This whole approach can also be utilized online tool by different other methods also of Ramachandran plot of structure validation. So, here one online tool or programmatically also Ramachandran plot analysis validation can be understood. So, in previous lecture when we understood modular through the Python script, that same thing can be utilized, or through expassi.org also we can go and we can do Swiss structure assessment. In Swiss structure assessment, we can upload our structure and the same structure validation can be done through Ramachandran plot validation. When we start doing Ramachandran plot validation through this Swiss structure is using ExPASI. What is ExPASI? Expert Protein Analysis System, in which different methods are being reported. Here the Ramachandran plot and SAVS analysis will be done for validating different programs, and they can give us better pictorial representation, same like how SAVS Procheck gives. It gives us 3D profile data and amino acid sequence structure information in graphical representation. The segment lacking in quality can also be identified and a residue plot can be represented. Under this SAVS, the next methodology which is need to be analyzed to understand the structural stereochemistry of the 3D structure protein is verified 3D and Procheck. What is Procheck? It is it understands the multiple, it takes consideration of multiple program specialized to check for stereochemical quality of the model, and for this it takes into consideration of RMS Z value and the Z score value. So, Z score value is nothing but the range of values which compile or which aggregates the statistical parameters same like p value. So, in Procheck or what if also we understand the steric hindrance of the 3D structure protein using stereochemistry. The third best method in SAVS is ERAT. As we have already seen Procheck, we have seen Procheck, which in the coming lecture Procheck will be taken as a separate explanation to explain the model validation using Procheck, but a brief explanation is given in the present lecture as well. In the third one is our ERAT. It is how well does the quality of the model structure with respect to atomic interactions. So, we can understand the atomic interaction of the protein under this. The ERAT has an atomic interaction followers certain, it understands the certain patterns, errors, identified the errors while modeling and during randomization. It is a statistical description of standard non-bonded interactions when fitted against with query model and can distinguish the different errors. The errors are nothing but all the stereochemical parameters which has been attached as a functional group or as a protein structure. So, this ERAT also explains all these atomic interaction understanding. So, in this lecture to conclude, we have understood about structure analysis verification server, and specifically after homology modeling or by threading or by ab initio method, the structure which we have identified need to be validated. So, for this in this lecture what we understood is the very prominent online database, that is SAVS. Full form of SAVS is Structure Analysis of Verification Server. For this, as conclusion, what we did was we went to UCLA online portal and there we uploaded the protein and understood the different Procheck, Verify 3D and What If, in which we understood specifically for Ramachandran plot and studied. So, I hope in this whole lecture we have understood the model validation. In the coming lecture, now we will see specifically what if protein structure validation, and we will also see specifically the pipeline of protein modeling, how we will do it up, and a very specific online tool, that is Geno 3D for structure validation. So, happy protein modeling, happy structure prediction. Thank you. So, very important, by understanding this whole lecture, we have understood the basic concept of Ramachandran plot. Ramachandran plot, let's have a revision of Ramachandran plot for this lecture, because this is basic concept for protein structure modeling and analysis. So, what was Ramachandran plot? Ramachandran plot is a concept of 4-quadrant analysis and it is based upon the secondary structure prediction method. And we also saw in a Ramachandran plot plot, yes, we can understand is the x axis and y axis. X axis is our phi angle and y axis is our psi angle. It gives us the torsional angle understanding. How does a torsional angle is been built? The torsional angle is built between CO-NH bond, and phi and psi angle is used for the rotation and winding of the protein. So, to understand the alpha helix, beta sheets and loops, and to understand the secondary structure, Ramachandran plot is given. So, in this whole theory portion, in this whole lecture, the base concept was Procheck, of which aligns between Ramachandran plot, and Ramachandran plot analysis was depicted in different graphical parameters. The first graphical parameter was the Procheck allowed region, disallowed region, generously allowed region, and the core red region. The second plot was for specific glycine and specific amino acid. The third plot was histogram plot, which was giving the dark line and the dashed line representation to understand the stability statistics of Z score and p value. So, this all gave the understanding to validate our protein in sense of secondary structure validation. Finally, we also took a glance of what if and IRAT 3D, which in the coming classes we will study separately. So, now we can say the validation of the protein is perfectly done. Now, to little more stabilize the protein, we can go for model dynamic strategy and stability using Monte Carlo or Monte Dynamics approach, which use different force field and threshold by different open source software also. So, this is a 360 paradigm to understand protein structure modeling and analysis. So, thank you so much for this lecture. Now, let us go ahead for the next class of Verify 3D and IRAT to understand the analysis.